You're listening to Conferences Online Allergy from Children's Mercy Hospital in Kansas City, Missouri. Today is December 11, 2023. Our topic today, Abbas Chapter 3, Leukocyte Circulation and Migration into Tissues. Our presenter is Dr. Nikita Raje. She's Chief of the Section of Allergy and Immunology at Children's Mercy Hospital in Kansas City, Missouri.
All right. Good morning, everyone. Again, let's get started. Hopefully, you guys had a good weekend and had some time to review this chapter. So we'll be talking about Chapter 3 from Abbas textbook, Leukocyte Circulation and Migration into Tissues. I'm Nikita Raje from Children's Mercy, Kansas City, University of Missouri, Kansas City, and today is December 11, 2023. Hopefully you can hear me well and see the screen. Stop me in between if you guys have questions, or feel free to reach out later with any questions. Here are my disclosures. Nothing relevant to the talk today. The learning objectives for today are to recognize the types of adhesion molecules and to learn the pattern of migration of various types of leukocytes. Couple of pre-test questions here. We'll come back to these at the end. Migration of leukocytes out of the blood into tissues mainly occurs in which type of vessel? And which of the following statements correctly describes the migratory behavior of naive T lymphocytes? Here's today's outline. Several topics here. We'll get started with overview of leukocyte migration. So, as we know, the movement of immune cells is constant. It's also highly regulated, and it's actually purposeful. It's not that the immune cells are just kind of moving from one place to another or just flowing through. There is a purpose to it, and that purpose, based on that purpose, the movement is very, very regulated. So we want to know how it's regulated and why it's regulated. Few definitions as we go along. So you'll hear these words, migration or recruitment, and they pretty much mean the same thing. It describes the general process of leukocyte movement from blood into the tissues. There's also a term called homing, and the migration of leukocytes out of the blood circulation and into a particular tissue or site of infection is called homing. So you'll hear the description as being the neutrophils homing to the gut, or any particular tissue that needs that attention. Recirculation is another term. It means ability of lymphocytes to repeatedly home. So repeatedly end up in a tissue or site or a particular site, and that ability to do that repeatedly to secondary lymphoid organs is called recirculation. The lymphocytes reside for a short time in a particular lymph node and then returns to circulation to repeat that process again and again. So it's called recirculation. So where do these leukocytes move from one place to another? And it depends on the particular type of cell. So if it's a myeloid cell, it's produced in the primary lymphoid organ, but then it moves to blood circulation from the site of production, which is bone marrow, to the blood circulation, and from there it can go into the tissues when needed. And that's all. Those are the sites where you'll find the myeloid cells. Versus naive lymphoid cells, naive means that they are mature lymphoid cells, but they haven't come across an antigen that they can interact with. So these are produced in the primary lymphoid organs, then they go to the blood circulation, and then they go to the secondary lymphoid organs. As we talked about the recirculation, that's where these naive lymphocyte cells are going to go. Effector lymphoid cells, on the other hand, are produced in secondary lymphoid organ, and we'll talk about how that process occurs. Then they move to blood circulation, and then they go into the tissues. A few principles of migration are that the cells need temporary adhesion. The leukocytes will have temporary adhesion of the molecules on them, and those adhesion molecules will interact with some other molecules or cells, such as endothelial cells of blood vessels or tissue cells. So endothelial cells express adhesion molecules again that interact with the adhesion molecules on leukocytes. They also express chemokines, whereas the leukocytes express adhesion molecules and chemokine receptors so that they can interact with the endothelial cells. So now let's talk about the adhesion molecules of leukocytes and endothelial cells that are involved in leukocyte recruitment. These are called selectins and selectin ligands, as well as integrins and integrin ligands. So we'll start with the description of selectins and selectin ligands. So the word lectin itself means it's a surface protein that can bind to a carbohydrate. That's all it means. The lectins that are specific for adhesion molecules, so a carbohydrate binding protein adhesion molecule is called selectin. Selectin ligands, as the word describes, it's a ligand for the selectins. These are sialylated carbohydrate groups of Lewis X and Lewis A family. They have a core sialylated and fucosylated moiety in them, and they are present on glycoproteins of various cells. So the sialylated and fucosylated moiety is really important, and we'll come back to that concept. But these are glycoproteins that are going to be able to bind to the selectins, right? So these are called selectin ligands. The function of selectins and selectin ligands, or their interaction, is to provide low affinity adhesion. So there are different types of selectins: P-selectin, E-selectin, and L-selectin. The CD marker for all these selectins is CD62. And depending on what type, you'll see the letters of CD62, P, E, or L. So P-selectin is present on the endothelium, and it can bind to its ligand, which is sial-Lewis X, on PSGL, so glycoprotein. That ligand is present on neutrophils, monocytes, T lymphocytes. So as you can see, the selectins are on endothelium here, and the ligand is on the cells. E-selectin is similar. E for endothelium, so it is on the selectin. E-selectin is on endothelium, versus the ligand is again on the cells. Compared to those, L-selectin is the one where it's present. The selectin itself is present on the cells. It's called CD62L, and its ligand is present on the endothelium. So various ones that you'll see here, ligands for selectins are glycam-1, CD34, medcam-1. So there are several others of ligands that can bind to selectins. So keep that in mind, and then we'll come back to how they work, right? So now we talk about integrins. Integrins are a little different from selectins. These are heterodimeric proteins. What does that mean? It's dimeric, so there are two molecules. These are two chains, and they are heterodimeric, which means the two chains are different. So they contain two non-covalently linked polypeptide chains. These are either alpha type or beta type. So there are 30 different integrins made out of 15 types of alpha chains and 7 types of beta chains, and various combinations between those. The extracellular head of these integrins, or these two chains, bind to the ligands for the integrins. And then the intracellular domain interacts with cytoskeletal components like actin and tropomyosin. Once the ligand is bound to the extracellular domain, the intracellular domain integrates signals with the cytoskeletal dependent, basically signals so that they can give rise to motility, change in shape, and maybe phagocytic responses for these cells. So the second half of the table here, integrins, there are again various integrins like LSA1, MAC1, VLA4, alpha 4, beta 7. And if you see, there are two chains. So the alpha chain and the beta chain. So the alpha chain is CD11A, CD11B, there is CD11C, and they can interact with the beta integrin chain called CD18. So that's your LSA1 or MAC1. These integrins are mostly present on the cells, and the ligands are present on the endothelium of various tissues. And the ligands can vary based on the tissue we are talking about. So again, for second years, memorize the name of the integrin along with the CD marker names, and we'll come back to that. The integrin ligands are also known as CAMs, so intercellular adhesion molecule. So ICAM is intercellular adhesion molecule or vascular cell adhesion molecule. So the CAM stands for cellular adhesion molecule. All right. So those were the two different types of adhesion molecules. Now we talk about another one, another kind of molecule involved with circulation and migration, which are called chemokines, and their receptors, chemokine receptors. So what are chemokines? These are type of cytokine that have a very specific role. So it's a family of structurally homologous cytokines that stimulate leukocyte movement and regulate the migration of leukocytes from the blood to the tissue. So that's the purpose for chemokines. So chemokines have various families. They are divided into various families based on the number of cysteine residues in them, as well as the structure of where the cysteine residues are located. So a family with CC means that there are two cysteine residues that are next to each other. These are called beta chemokines, whereas CXC family has X stands for any other amino acid. So if there are two cysteine molecules or residues with one amino acid in between them, these make up a family of alpha chemokines. There's also a family with single cysteine residue, and there is another family called CX3C, which means there are two cysteine residues with any other three amino acids, which can vary. So all these are families CC, CXC, C, and CX3C are different families of chemokines. CC and CXC are the chemokines that are produced by leukocytes and tissue cells. And these chemokines are produced under the influence of inflammatory cytokines, or because there was recognition of microbes through various innate receptors. The CC type of chemokines are produced by activated T cells, and they form a link between adaptive immunity and recruitment of inflammatory leukocytes, which means the innate immunity. So these chemokines are going to bind to their receptors called chemokine receptors. And as you see in that figure, this is a 7-trans membrane, which means it spans the cell membrane 7 times. It's a 7-trans membrane GTP binding G protein coupled receptor. So it belongs to the GPCR superfamily, and it helps in initiating an intracellular response through the G proteins. Because of that signaling, there will be stimulation of cytoskeletal changes, polymerization of actin and myosin filaments, and that helps with the motility of the cell. It also increases the integrin affinity for the ligand, and we'll talk about that in a little bit more in depth. So the chemokine binds to the chemokine receptors, help in the migration or motility of the cells, but also keep in mind that as the chemokine is exposed to the chemokine receptors, the receptors get downregulated. So there is negative feedback. So just like the chemokines have chemokine families, their receptors also have similar families. So CCR, which means these are the receptors for CC chemokines, Uh, they are numbered, so CCR1 to CCR10. CXC receptors go from 1 to 6 that we know of yet. CX3CR goes with pretty much just one chemokine receptor that goes with one chemokine. So CX3CL1 is the chemokine that binds CX3CR1 receptor. Again, the receptors are present on various cells. So here is a list of these chemokines and their receptors, right? So these various families, CC chemokines, which are the most abundant. As you can see, there's a long list. Like everything else in immunology, these chemokine receptors are very well described with their numbers, but they also have their second name that's more descriptive. And some of these are really important in how the cells function or where do they go. And so, for example, eotaxin, TARC, you guys are well aware of these, some of these chemokines that are important for in allergic diseases. So recruitment of eosinophils, basophils, Th2 recruitment for eotaxin. Similarly, TARC is another one that's important, helps in T cell recruitment. There is CCL2, CCL3 that might be for a lot of different leukocytes. CCL4 again for several other cells as well. CCL19 and 21, as you would know, bind to CCR7 that we talked about last chapter. How CCR7 is present on cells that are attracted to places where CCL19 and 21 are produced, which is the T cell zone of secondary lymphoid organs. So this association is important to remember. Similarly, memorize that CCR9 goes to the place where CCL25 is produced. So cells that have CCR9 will go to a place where CCL25 is produced, which is in intestine. Similarly, CCR10 can go to a site where CCL27 is produced. So those cells with CCR10 will go to skin. And this helps in lymphocyte recruitment to intestine or skin, as these receptors lead them to. Similarly, CXCL8, CXCL1 are important for neutrophil recruitment. In general, think about CXCL8 as IL-8. It's an interleukin 8 that helps in neutrophil recruitment. So on and so forth. So you can go through that list, but some of the important ones that I really wanted to make sure that you know the names for. CCL5, for example, RANTES. You have to know it's an abbreviation for a particular type of chemokine, what it does. So I'll let you guys read that. All right, biological actions of chemokines. So now we talk about how these work. As we know, chemokines, I had described that basically these are molecules that help in recruitment of leukocytes. They also help in development of lymphoid organs and regulate the traffic of lymphocytes, other leukocytes through the lymphoid organs. They help in migration of dendritic cells from the site of infection into draining lymph node as well. All right, so this is a concept that's really important to understand. So we talked about selectins, selectin ligands, integrins, integrin ligands, and chemokines and chemokine receptors. Now they have to know how there is interaction between those molecules. So if you think about signaling, typically the concept is that the ligand binds to its receptor on the outside, and that leads to signaling on the intracellular or cytoplasmic side. And so there is effect intracellularly. That's the general concept. So when it comes to integrins, there is also something that's called inside-out signaling, which means that there is some activity inside the cell that leads to signaling, resulting into a change on the outside of the cell. So that's the concept. And so how does that happen? So this inside-out signaling means it's an ability to respond to intracellular signals by rapidly increasing their affinity for their ligands. So thinking about selectins, and selectin ligands, when they interact with each other, the cell starts to roll on the surface. However, once that starts to happen, along with that, there are chemokines that are going to bind to the chemokine receptors. When that interaction occurs, there is signaling through that GPCR, right, receptors. And when that happens, there is interaction between those molecules with the integrins. So that signaling of chemokine and chemokine receptors leads to change in the integrin affinity. So if you see in this figure here, initially there is low affinity interaction between integrin and its ligand, so ICAM. There is low affinity interaction. However, after the chemokine receptor interaction, there is inside-out signaling so that the affinity with which integrin binds to its ligand increases. So there is conformational change that leads to increased affinity. So that's called inside-out signaling. All right. Another thing to remember is chemokines induce membrane clustering of integrins that also increases the avidity. So the affinity increases. Avidity is the overall interaction between various integrins binding to their ligands on the from the leukocytes to the endothelium. So that interaction overall increases the avidity as well. There are some atypical chemokine receptors. So ACKR stands for atypical chemokine receptors. They do not engage the same kind of G protein signaling pathways. There are 4 of those, and they bind to most of the inflammatory chemokines. One example is ACKR1 or DARK that's present on erythrocytes. The signaling is through a pathway that is dependent on G protein regulatory proteins called beta arrestins. All right, moving on. So we learned about the different molecules that are involved in migration and circulation. Now we want to look at those interactions on how that migration occurs. Sorry. All right. So let's look at this figure. So say there is an infection somewhere. At the site of infection, there are those innate cells like macrophages, dendritic cells, mast cells that are going to release cytokines. Those inflammatory cytokines are going to reach the circulation and tell the cells to come to the site of infection to help. And that signal, or basically message that reaches the circulation, leads to a series of events that helps the leukocytes to move from the circulation, come out of the circulation, and go to the site of infection. So think of, say you guys are on call all the time. Think of someone, a physician on call who is, and at the end of the day, going home and going on the freeway and being paged to, and once you answer the page, you are told that you are needed back for an urgent situation. In that case, you would have to move from the freeway or from a fast lane, go to the slower lane, kind of apply brakes and come out of the freeway and go back to the site where you are needed. Same thing with the neutrophils. I'm going to take neutrophils for an example. This happens with all the leukocytes, but think of neutrophils that are on call 24/7. They are flowing through the fast lane of the circulation, right, all the time, 24/7. They are on call 24/7. So when they are called upon by these messages saying we are needed, we need help, the neutrophils realize that they are needed and they start the process to move out of the circulation. But remember, they are flowing in the blood flow in the fast lane. So they have to really come to closer to the cell wall, which is the slower lane of the circulation. As they move there, because of these messages that they get, they start to express the selectin ligands on their surface that can interact with selectins that are present on the endothelial wall. As they interact with those selectins, they start rolling. So they slow down and start rolling because they interact with selectin ligands. The selectin ligands start to interact with the selectins on the endothelial surface, so that it's almost like attachment and then leading to the detachment of it pretty quickly, and then it attaches to another selectin as it moves along the surface. So it starts rolling on the surface of the endothelium. As that happens, the integrins also interact with their ligands on the endothelial surface, and that actually slows it down further, but that's a lower affinity in interaction. Then comes along the chemokine that binds to the chemokine receptor, and that improves the affinity of integrin and integrin ligation. Overall, at the end of this process, the leukocyte comes to a complete stop. It applies brakes, basically, and comes to a complete stop on the surface of the endothelium, after which it can migrate in between the cells or through the endothelial cells. So that's called transmigration. So either through the cells, so transcellular or paracellular, which means between the endothelial cells, and they pretty much almost make a space in between the cells by using their adhesion molecules that includes PCAM or CD31. Once it comes out of the circulation, the chemokines attract the cell so that it knows where to go. It follows a scent of chemokines to go to the site of infection. All right, so what happens if the leukocytes have a problem with the adhesion molecules, any of these molecules? You have heard about leukocyte adhesion defect. There are 3 different types. LAD type 1. Does anyone know what molecule is affected, what adhesion molecule is affected with LAD type 1?
Is this with like CD18? I think this affects the integrin portion, right?
Correct. So CD18 defect. So if you remember the table with LFA1, MAC1, CD11A, CD11B interact with CD18 to form integrins. And if CD18, so the beta subunit of LFA1 and MAC1, if that's affected, you get what is known as leukocyte adhesion type 1. LAD type 2, anyone know what's affected here?
I would say.
Go ahead, Sean.
Okay. SLEX?
Yeah, what does that stand for? So maybe I didn't mention. So it's the selectins, or so the Sil Lewis X is what's affected. So they lack. So selectins bind to selectin ligands. Selectin ligands have these sialylated bodies, right? Sil Lewis X is one of the important ones. So what happens with LAD type 2 is there is lack of Golgi fucose transporter. So fucosylation is affected because it's needed for the expression of selectin ligands like Sil Lewis X. So because of that, these patients with LAD type 2 have problems with interaction between selectins and selectin ligands. So LAD type 1 is an integrin problem. LAD type 2 is selectin problem. What's LAD type 3?
It's the inside-out signaling affected.
Yeah, so inside-out signaling is affected, which means chemokine binding to its receptor and that interaction leading to improved affinity of integrin. That's inside-out signaling, and if that's affected because of defect in something called kindlin 3, gene defect in kindlin 3, that causes adhesion defect type 3. So we're going to move on, but I would like you guys to learn more about LED type 1, type 2, type 3. The typical LED, you hear about high neutrophil count and delayed separation of umbilical cord, wound healing issues, where if there are skin subcutaneous infections, if you try to drain them, there's no pus formation. All of those are features of LED type 1, which is the most severe. LED type 2 is a defect with more of growth and developmental issues, and LED type 3, where there are platelet issues. So the most severe one is LED type 1 that you typically talk about, but you have to know about these LED type 2 and type 3 as well. All right, moving along.
Quick question, Dr. Raje.
Yeah.
Are all 3 types autosomal recessive?
They are all rare. Among them, LED type, so their presentation is different. Even though they are all autosomal recessive, they can be pretty severe. LED type 1 is the most severe, where it presents pretty early in life, versus LED type 2, type 3, they can present later in life. So the infection part is way less severe in type 2 and type 3 compared to LED type 1. So they present differently, but I think the genes are still autosomal recessive. And I might have to check on that, the third one. I think they are all autosomal recessive, but I don't know if there have been newer variants in those genes. If any of them can be inherited autosomal dominant fashion. That's a good question. So definitely we'll check on that. All right, moving along, we are going to be talking about specific cells now. So we talked about the general process, and this is almost going to be a repetition of what we talked about, how these cells move, but maybe more specifics for each of these cells. So first we start with neutrophils and monocytes. So pretty much all phagocytes, right? And as we know, these are myeloid cells, so they move from primary lymphoid organ to the circulation to the tissue. That's their trajectory of where they go from one place to another. So neutrophils, as I mentioned, are first responders, right? So they interact with, there will be selectin ligand interaction, there will be integrin interaction, and then there is chemokine and chemokine receptor interaction. The chemokine important for neutrophils is IL-8 or CXCL8. Same thing, 2 names for the same molecule. It's produced by macrophages at the site of infection, telling the neutrophils to help. And the IL-8 reaches the circulation and sends the message to the neutrophils as it interacts with the CXCR1, CXCR2, and those cells, which are neutrophils, are going to get the message to move out of the circulation and go to the site of infection. Compared to neutrophils, monocytes are late responders. Here, the chemokine CCL2 or MCP1 is important. It binds to its receptor CCR2. And because of the CCR2 bearing cells, monocytes get the message saying they are being recruited. They are going to move out of the circulation, go to the site of infection, and in the tissue, they become macrophages. There are also non-classical monocytes instead of the CCL2 or MCP1, which is an inflammatory chemokine. This can bind to, so the monocytes with non-classical phenotype have CX3CR1 receptor on them that will bind to the chemokine CX3CR1. All right, so now we move on to talk about T lymphocytes and their migration. So we know that the T cells that are produced in the bone marrow, they start out as naive T cells. These are mature T cells, but they haven't come across an antigen. So if you think about a cell, lymphoid cell that's produced in the bone marrow, It enters the circulation, and then from the circulation, it comes out into, it enters the, say, a lymph node through high endothelial venule, right? So here's our naive. I hope you see my pointer. If you don't, let me know. But here is a lymphocyte, naive lymphocyte that entered the lymph node, and it's gonna go through the lymph node, move through the T cell zone, and come out of the circulation and keep recirculating to another lymph node, and that keeps on going till it comes across the antigen that it can interact with. So say there is an infection, and at that site, there are macrophages and other sentinel cells that produce some inflammatory markers, and as a result of some of the interactions, say the dendritic cells are the ones that pick up and process that antigen. Those dendritic cells are going to enter the lymphatic tissue and bring the antigen to be presented to the lymph node. That antigen is going to be presented to the naive T cell, and if it can interact with that naive T cell is going to now be an activated T cell. Once it's activated, it stays in the lymph node for longer, and then when it comes out of the circulation, instead of recirculating, now it's going to go to the tissue where it's needed. Rather, the purpose of the these cells to recirculate from one lymph node to another is to search for the antigen it can interact with. If it finds that antigen, then it no longer needs to recirculate. In those cases, it can move from the lymph node to the periphery where they are needed. So it's purposeful movement, and based on the purpose, the trajectory changes. So a naive T cell will go from one lymph node to another and continue to recirculate till it comes across the antigen that it was looking for. Once it comes across the antigen, it's no longer a naive T cell, it becomes an activated T cell. It's retained in the lymph node for a little longer time. It goes through proliferation, then undergoes differentiation into an activated T cell, and that activated T cell will make effector cells that will go to the site of infection to help. We know that the lymphocytes enter the lymph nodes or secondary lymphoid organs through HEVs. Here is just a picture, different types of pictures of HEVs. The development of HEVs requires cytokines such as lymphotoxins. All right. I believe we talked about the process of recirculation, but something to remember, the recirculation in terms of, so say there is tissue inflammation. It increases the blood flow to the lymph nodes. That increases more T cells to come into lymphoid organs. And why is that important? Because you want those lymphocytes to be able to come across the antigen that is coming through due to that inflammation. During that time, as the blood flow increases, the inflow is increased, but the egress of the lymphocytes is decreased. So the T cells are retained in the lymph nodes for a longer time. So one, it has a higher chance of meeting the antigen. The antigen that a particular T cell can bind to is called its cognate antigen. And if it comes across that antigen, it needs time to interact with that antigen for longer time to go through that differentiation process. So during inflammation, the influx of cells to the lymph node increases, the egress decreases so that the T cells are retained for a longer time. All right, so let's go through that process of selectins, integrins, and then chemokine interactions for T cells. So say the naive T cells come in, right, in the HEV, how do they know where to come out? So the L-selectin or CD62L on the naive T cells can bind to the selectin ligand on the high endothelial venule. Say for the lymph nodes, that selectin ligand is called peripheral node addressing. And as the name suggests, it's pretty much giving that address. So L-selectin is just looking for its buddy, so selectin ligand, L-selectin's particular ligand for the lymph node is called peripheral node addressins. Now, different places in in different tissues, the peripheral node addressin can be different. Which means, so for example, the lymph nodes that are in the gut might be different from lymph nodes that are looking for cells with L-selectin in another tissue. So typical ones, peripheral node addressins, are glycam-1 and CD34. They can bind to their selectins or integrins and attract more cells, naive T cells that have L-selectin on them. Versus in peripheral tissue, Peyer's patches in the gut can bind to MADCAM1. So MADCAM1 and glycam-1 are a couple of peripheral node addressins that attract cells that have L-selectin on them. Once that selectin-selectin ligand interaction occurs, the next interaction is integrins with their integrin ligand. So integrin ligand is LFA1, to which the integrins on the T cells, LFA1 will interact with ICAMs so that there is a firm adhesion between the T cells and the endothelial wall. And then the chemokines that are produced in the T cell zone of the lymph node, which is CCL19 and CCL21, attract cells, naive T cells, because they have CCR7 on them. The cells that have L-selectin and CCR7, those T cells are called naive T cells, and they exactly know where they are going. They are going to come out of the HEV because that L-selectin is bind to the peripheral addressin, and then they are going to be moving towards a zone where that CCR7 can bind to the chemokine receptor. CCR7 can bind to the chemokine that's produced in T cell zone. Give me 1 second, I'll get water, sorry. All right, sorry about that. Okay. The table here pretty much gives you the same information, pretty much showing you how there is interaction between the various adhesion molecules for naive T cells and compare them to activated or effector T cells. The L-selectin for naive T cells versus the E and P selectin for the activated T cells. And then you see some of the other interactions as well. Overall, the figure here basically shows you how those cells use up their adhesion molecules to interact with their ligands, and then once they come across the antigen that they are specific for, they are going to be activated, and then the activated or effector T cells are going to go to take them to the site of infection. So how does the T cell know, right? Like the blood flow itself can only keep the T cell in the lymph node for a little short period. But these T cells, once they are active, they need more time to spend in the lymph node. So how do these cells know which way to go? And the reason for that regulation of their movement is a gradient called CYP gradient. CYP stands for sphingosine 1-phosphate. and CYP binds to its receptor called CYPR1. This receptor is present on the T cells. So the rule is that the cells move from cells with this receptor move towards the gradient. So if there is high concentration of CYP, like in blood and lymph, the CYPR1 can interact with that, but then gets internalized. So then it moves towards the gradient, whereas if there is low concentration, for example, in the tissues, there is an enzyme called CYP lyase that gets the CYP broken down, because of which there is low concentration of CYP there. In those situations, there is re-expression of CYPR1 on the naive T cells. So basically, when there is a naive to the lower concentration, whenever they have the CYPR1, they are moving towards the place where they can find CYP. So naive T cells move towards the lymph node, and in here, when they are inside the cell and they get activated, they start expressing something called CD69. CD69 binds to the CYPR1 on the cells, and so they become insensitive to the CYP gradient. So instead of moving out of that high concentration and then move out of the lymph node, they become insensitive to the gradient, so they are retained in the lymph node for a longer time. So those activated T cells are going to stay there, undergo expansion, proliferation, and then differentiate. Once they differentiate into effector T cells, they actually lose their marker CD69. Once they lose CD69, there is increased expression of CYPR1 on their surface. So now they become sensitive to the concentration gradient, so they start moving out of the lymph node and they can exit the lymph node. Now there is an immunosuppressant that's used with this property. So it's called fingolimod, and the fingolimod is something that can bind to the CYPR1, and so it will retain those lymph T cells that are treated with fingolimod in the lymph node for a longer time, so they don't come out. If they're not coming out of the circulation, they're not going to cause the inflammation that they would typically cause. So it's used as an immunosuppressant.
Can I ask a quick question? Oh.
Yeah. Mm-hmm.
So I read this multiple times yesterday trying to understand it. So I guess in my mind when I'm thinking, so essentially if it's a naive like T cell and it's in circulation where there's going to be low CYP gradient because of the enzyme that's breaking it down, it's going to go towards the lymph node so that it can theoretically interact with the antigens, right? So when it says internalization, does that mean that it's endocytosed, or what does that mean?
So it goes in.
Okay,
so it's no longer available to
so it's
interact.
no longer available, but then how is it that when it does interact with an antigen and becomes activated for a T cell, where does the interaction of the CD69 and the internalized receptor occur? Is that intracellularly that allows that to stay within the lymph node, or I think that's where I'm getting tripped up.
So are you trying to figure out how the CD69 interacts with the receptor? Because CD69 is just causing the re-expression.
Oh, so it causes the re-expression of the CYPR1, so then it can stay within the lymph node. And then it can interact. Okay.
So any cell that has, we are talking about T cells, but theoretically, any cell that has CYPR1 on it is going to be able to bind to CYP, right? And so wherever there is CYP, basically the T cells are going to stay there because the receptor is binding to that CYP. So when it's activated, it can bind to the CYPR1. When the when the when the CYP-R1 is internalized, there is nothing for the CYP to bind to. Right? When it's internalized, even if the CYP is there, it doesn't do anything. So it becomes insensitive. So it's not going to move along that gradient. It doesn't matter where the CYP is. That cell is going to stay there. When that CYP-R1 comes out is when it's going to follow the gradient. And the gradient is from high concentration to low concentration. So it moves out of there to go to the low concentration site. So when they lose their CD69, there is re-expression of CYP-R1 so that it can get out of that lymph node. Does that even answer your question? Sorry. Maybe, and there might be another way to describe this. So
I think I understand it better. I think it's just that component of thinking about like after internalization and then the expression of CD69, that interplay with the S1PR1 to allow it to stay before it like loses the receptor again and then exiting the lymph node. I think it's just that piece, maybe the re-expression part that's confusing me.
Right. And so basically, the CD69, whatever function that CD69 does, fingolimide does a similar thing, right? Because when it expresses, a cell expresses CD69, it's going to be retained in the lymph node for longer time. In that case, in the natural circumstances, it's an activated T cell that just needs more time. It's buying time to spend in the lymph node longer so that it goes through the whole process of differentiation. On the other hand, when fingolimod does that, it basically is binding to CYP-R1 and it's retaining the lymph nodes in the lymph node for longer, so it's blocking the egress of T cells. And the T cells have to come out to do their job. So if you give them fingolimod, the T cells just stay in the lymph node for a longer time. So that's the purpose of using it as an immunosuppressant. All right, let me know if you have more questions about that, though. I know this is definitely confusing, yeah. All right. So other lymphoid organs, we were talking about lymph node for the most of the time, but thinking about any cell that's going to gut lymphoid tissue, so Peyer's patches, mesenteric lymph nodes, they're still using similar process of selectin binding to selectin ligand, helping in rolling of the cells on endothelium. Then the integrins come along, then the chemokines come along. Typically, the naive T cells, they are gonna interact with this particular addressin called mucosal addressin cell adhesion molecule, or MADCAM1. MADCAM1 can bind to the selectin. It's interesting because it can also be an integrin. It's a CAM, so it can be an integrin ligand as well. And so it can bind to L-selectin and it can also bind to the integrin alpha-4 beta-7. So that interaction of alpha-4 beta-7 with MADCAM is a really important one. It helps in bringing those cells to a complete stop so that they can come out into the patches from the circulation. So it helps in homing. This interaction of MADCAM1 alpha-4 beta-7 helps in homing of these cells to the gut. When we talk about spleen, there are no HEVs. There is no selectin integrin mechanism, right? In some of these, particularly in spleen for some of these cells. But the CCR7 binding helps the cells to know where they are going. So if these are, say, developing B cells and they express developing T cells that are going to the spleen, when they express CCR7, they know to go to the white pulp. So in that white pulp, the T cell zone will have the lymphocytes going to the T cell zone, right? CCR7 going to CCL19, CCL21. But then the B cells will have interaction with the other chemokines and chemokine receptors that pull them into the follicles. Regardless, the CCR7 is a basic thing that will pull them towards the white pulp. All right, now we talk about effector T cells. For all along, we were talking about naive T cells and how they go to these places. Now we are talking about effector T cells going to the site of infection, right? And so once the cells are activated and differentiate into effector T cells, their expression of CCR7 and L-selectin goes down. And it's very purposeful so that they do not move to the lymph nodes, because anything that expresses CCR7 is going to be pulled to another lymph node. So now the expression of CCR7 and L-selectin goes down, and so they can move along the circulation and they can start expressing more selectins and integrins and chemokine receptors that take them to the periphery and to the peripheral tissue that needs them. All right, we talked about this, so this is just a repetition. Gut homing or skin homing. So for gut homing, there is CCR9. Any cell that has CCR9 is going to be attracted to the gut because CCL25 is produced that. So cell bearing CCR9 is going to be attracted to the gut where CCL25 is produced. That's where the MERDCAM1 binds to alpha-4 beta-7 integrin, so that it will come out of the endothelium. Those cells are going to come out of the endothelium in that region, HEVs in that region, so that they home to the gut. Another chemokine receptor is CCR10 that interacts with the chemokine CCL28. So remember CCL25, 28, they are both present in gut. CCL28 is also present in lung. And cells with CCR9 will go to CCL25, CCR10 will go to CCL28 rich place. Similarly, in the skin, there are cells that express cutaneous lymphocyte antigen or CLA. That is a selectin ligand that can bind to E-selectin. So the cells that have CLA on it will bind to the endothelium with E-selectin in it, which is in the skin. Also, cells with CCR4, 8, 10 will be attracted towards chemokines CCL17, CCL1, CCL27, where they end up in the skin. There is a drug called, so basically it antagonizes alpha-4 beta-7, vedolizumab, that is used in early onset IBD or in general IBD because it prevents the inflammatory cells from entering the gut. And the last one is about T cell and memory T cell migration, and then B cell migration. So the memory cells, again, instead of CD45RA, they express CD45RO. Central memory T cells go to the lymph nodes, so they express CCR7 L-selectin on them. Effector memory T cells have low levels of CCR7 L-selectin, so they do not go to lymph nodes. They go to the tissues. B cells go from bone marrow to the circulation, to the spleen or other secondary lymphoid organs. So again, in the spleen, they'll use CXCR5 that helps them to move towards the B cell follicles where CXCL13 is produced. For the lymph nodes, we know that they express CXCR4 that binds to CXCL12 and CCR7 that binds to 19 and 21. Follicle entry requires CXCR5 that binds to CXCL13. And then Peyer's patches, again, similar kind of interaction. Plasma cells in lymph nodes and spleen home to bone marrow and tissue sites. So they move from one place to another. And then follicular B cells in the spleen will migrate to the marginal zone and then carried through the red pulp and into the circulation. CYP1 deficient B cells have diminished ability to leave the spleen. So again, same kind of interaction and CYP gradient for B cells as well. And then in general, for the migration of plasma cells, you see there are certain types of chemokine receptors and integrins, but pretty much mucosal homing, alpha-4 beta-7, or chemokines that help them to get to the mucosal endothelial cells. whether it's IgG secreting, plasma cells IgA secreting, depending on where they are going, they'll interact with those kinds of chemokines. All right. I think this is the summary of what we talked about. And then let's go over a couple of questions. Migration of leukocytes out of the blood into the tissues mainly occurs in which type of vessel?
Should be venules.
Yeah.
Okay, venules is correct. And then which of the following statement correctly describes the migratory behavior of T lymphocytes? This is pretty descriptive. Are they going from blood to the lymph nodes or into tissues, from thymus to lymphatics, stationary?
I think this is A.
That is correct. Going from blood to lymph nodes and then back into the blood via lymphatics. All right. That was the end of the chapter today. Feel free to reach out with any questions. And then the next chapter is Jeopardy.